In the introductory lecture that I gave last week, I commented, that one of the intents of CHEM 311 is to help the students progress towards independence as chemical professionals. And so one critical component, therefore, of CHEM 311, is a research project, where students are gonna ideate, come up with a research project themselves and read journal articles, so that they can develop the hypothesis, but also develop, their protocols and then execute that in the lab and analyze the data that they obtain and then write a report and present it, to the class. So in this lecture today, I'm gonna attempt, to give you some more details on what is expected of that research project that's gonna account for 20% of your final grade. Now, how do you select the research topic? Maybe first of all, I'm gonna give you some bit of precedence of types of topics that previous students in Chem 311 have done. For example, I think it was 2 years ago or so, this was a list of 3-4 years ago, this was the list of topics, that some of the students, had done for the research project. For example, an on and off switch for triglyceride hydrolysis, based on responsive polymers. It's an excellent topic that was done previously before. Another one, a lactose biosensor, based on the use of redox responsive polymers. Again, students, developed the responsive polymers. This is what people call smart polymers that are very selective, towards certain types of chemical species based on the way, they are tailor-made. And of course, as a mentor, I would help the students towards that end. Maybe another interesting project, that was done a couple of years ago is a THC, tetrahydrocannabinoid, molecularly imprinted sensor. It's an electrochemical sensor, for detection of THC, sort of to address the the crisis, on the use of, some of these substances. Maybe another compound or other project, that was done before is an inexpensive sensor for detection, of E. coli. I think last year, this was the topics that students selected from, and you're gonna see they might bear relationship, with the types of topics, you guys might select. Maybe a good example is a solid phase microextraction GC platform, for detection of cortisol and ketones in human sweat. And of course, you're gonna see all these projects first start, with identification of a societal problem, and then we ideate, how we can use analytical chemistry to design probably something like a sensor that can be used to address that societal problem. And then so, the way I think innovation should happen, which is what we are trying to train you here, the ecosystem or the art of innovation, is you start with a problem that the society cares about, and then you use your analytical, technical skills, to solve that problem. So another interesting project, that no one picked it up, but it's interesting, is microneedle, sensor that can be used for detection of fruit ripening, mushroom quality assessment and olive oil analytics. It's an interesting project. I think a student last year did this project, methamphetamine or crystal meth detection, using a polymer, that is very, very selective, to capture on the crystal meth, and then detect it using, I think she used electrochemical detection in her end. Maybe another interesting, project, that was done is a biomass water remediation filter using waste. Again, that was an interesting project that was done, and that still remains going on in my research group. Maybe last one just to mention, a wearable microneedle patch sensor for detection of food quality. Again, we are dealing with issues on food spoilage and wastage. It's a big problem, in Western countries especially. And so can we develop sensors that can use to monitor the quality, of the food, so that, the food is not wasted. So lots of problems, in the society out there, that we can use analytical chemistry actually to solve. Now, I should comment, that we do not just do these small projects, and then the CHEM 311 students leave it at that, but actually students have published, some of their results in published peer reviewed journal articles. Now, which I would say is possibly the climax, of, academic research. Now, if you go to graduate school, this is sort of what the masters and the PhD students, would aspire to do. They do, the research in the lab, and they write the journal articles which they publish. And what by doing so, you're advancing, the chemical knowledge, to the scientific community, but also, of course, to the public. And so I hope, that actually inspires you, to know that it's not just a small project, but it can go beyond, just a term paper, and you can publish the work, in a peer-reviewed journal article. And by doing so, actually, you create for yourself a tremendous mileage. If at all you go to graduate school, it's in most cases it's very, very rare to find undergraduate students having published journal articles, and so you get a lot of a leg ahead in terms of possible selection, for graduate school programs. But even if you don't intend to go to graduate school and you intend to, go and get a job, having published, sort of a peer-reviewed journal article, shows, the excellence, of your work. And it shows, how independent you can be in critical thinking, and also solving problems. And it's also a classic example, of experiential learning. And of course, you go out there and everyone want to know what you can do. And by showing that you've published, a journal article is just, sort of an ambiguous, example, that you're an excellent student, with a good experiential, background. Now, maybe just to show you what students have published before, right from CHEM 311 projects, like I told you that THC sensing, a student did that work a couple of years ago, and that is a student, and they published the work in inorganic chemical communications. And, that gentleman now is working, in the industry here in Edmonton. And I must say, often I've seen, students who have shown that they have done some experiential learning opportunities in the university. Often they don't struggle actually, to find work out there. Maybe another excellent example of a published, paper coming from 311 students, is this one. Where the students develop, a chemiresistive sensor for detection of methanol, which is often used, to adulterate alcoholic beverages in different countries, because of course, methanol is gonna stone, people much more quickly, than ethanol. And so people unfortunately and scrupulously, take methanol and they spike it, in the brew, in alcohol. And then, of course, it becomes a problem. A lot of people have died, especially in some countries, Chile, in some countries in Africa as well. It's a big problem. And so again, this student here, and these are the student, and these are the students actually, from CHEM 311, they did this piece of work and they published it in the IEE census, an excellent journal actually. This gentleman, now has started his own company. And again, and that's the idea, training the students how to think, to innovate, and hopefully they don't just look for a job, but they can actually start, their own chemical technology companies. Maybe another paper that we published, with a Chem 311 students is this one, where we developed a sensor to detect resveratrol, which is an antioxidant, present in red wine, but also present in white wine, but it's more present in red wine in higher quantities. And these gentlemen took Chem 311, these gentlemen, these a lady as well, and they published that paper in an excellent journal called Analytical Methods. Now this person, works for EPCOR, here in the city, and I often invite him, into my classes, to give lectures, as I'm a K1 alumni, and maybe not this semester, because of the COVID situation, but nonetheless, those are the students who participated. This lady, again, she works in a company in BC, and this gentleman, I've talked to him already about him, he works for a company here in the city. Another paper again that came from Chem 311 student, it's a flexible imprinted sensor for detection of adrenaline, in sweat. An excellent paper actually, published, by this lady in Chem 311, and she works now, in the city, in a company in the city. Another paper that some of you might be interested in, especially those of you interested in microbiology, pathogens, and so forth. A pathogen imprinted hybrid polymer sensor, for selective detection of E. coli, and that one we just published early this year. And Wei Lu, who is currently one of my full-time research assistants, actually did that work, but she started the work actually in case of Chem 311 students, as well. Again, an excellent journal. I think this one is Medical Devices and Sensors journal. Maybe another example recently published, is a flexible sensor for detection of cortisol in sweat. This is a wearable device, and we could use, a smartphone, to monitor the amount of stress hormone, cortisol in sweat. And that was published, by these gentlemen who took Chem 311 possibly 2 years ago, and then took a Chem 498 a year after to continue on his project, and it was recently published. And it's generating a lot of actually interest, globally. I've gotten some some emails, from a professor in the University of Reading, who want us to continue to collaborate on this work and hopefully make a commercial device, out of this. And maybe lastly, this other paper published very recently, by Wei Lu. Again, I've just said, that she works with me currently as a full-time research assistant, but previously a Chem 311 student. And she published a paper on using microneedle sensors for detection of plant metabolites, such as polyphenols. And you're going to see in the topics, that I've given you, they're going to be somewhat an extension of this work, which is our own invention, and then, of course, you can see that it's got a very, very diversified possible applications in lots of different applications and areas. And so by doing that, again, we won't have a problem in terms of the execution of the work, because we've got all the expertise, that is needed in my research group, to help you guys on this. But again, a classic, I would say, invention, towards many different applications. So I'm recommending or proposing to you these different research topics under the theme microneedle electrode sensors, for precision agriculture and data analytics. So I propose, that students, can choose from this vast list of different topics, that they can do for their Chem 311 projects this semester. Maybe one project, the microneedle, and all of them actually are based on electrochemical detection. All of them are based on electrochemical detection. And you've heard me say, essentially, this is the future, because electrochemical sensors are very easily miniaturizable, and so you can make them very, very small, even into nanosensors, and that way, you can have them. It's almost an idea of democratizing chemistry, where the public, can actually do chemical analysis for various reasons, clinical diagnostics, environmental monitoring, in their homes, and so forth, which is the future really, of where analytical chemistry is going. So I propose maybe one student, can make a sensor for detection of humidity in soil, because you can see, that impacts things like the climate change and so on, water conservation, which is a big deal out there. You can also make, a wearable sensor to detect hydration in plants. That way we can only water the plants on demand instead of just watering the plants, and that way you waste the water. Another person maybe can work on detection of nutrients, such as nitrates in water. Another person can work on nitrates, nitrites in water using an electrochemical sensor. Similarly, another person can work on detection of phosphates, a big problem, actually, especially in feedlots, where you've got lots of cattle, for example, or animals that are producing a lot of waste, and there's runoff coming from those cattle feedlots, all that water going to the rivers, and it causes, eutrophication or an excess of nutrients, and then you've got problems with algae growth and microsystems and those kinds of problems. So it's a very classic, I think, project that is needed out there. Of course, these methods have been developed using conventional analytical methods, where people bring the sample into the lab and they use spectroscopies and so on, which are expensive techniques. But the idea with these kinds of projects is to package very small devices, that can be used in the field, where the problem is. Maybe another person, can work on phosphates also, but in this case, detecting them in soil, rather than in water. Maybe another person, can work in detection of pH, and so forth. Maybe even somebody else can work on detection of redox potential. Now, this one is very, very interesting, because redox potential is this bulk parameter, that can be used, for detection of plants, that are undergoing stress, be it from possible infection, or be it, from drought, or be it from lack of nutrients and so on. So it's a classical, clinical diagnostic indicator, for plants. And so I would really like somebody to actually work on that. And of course, we've done a bit of work on that in the lab, so it wouldn't be very difficult for the student who continues to do that work. For anyone interested in microbiology, again, somebody can work on developing a platform, that can deactivate or that can kill, bacteria or even other pathogens such as the virus, even this COVID-19. Actually, we had proposed that kind of a project recently with foreign, with a company here in the city. And so somebody can actually work on that, a platform that generates a disinfectant, and the disinfectant, can deactivate, the pathogen and so forth. Maybe another person, can work on something like this, making a platform that can release a pesticide, but only on demand. I think we know these days people spray pesticides everywhere, and it's a very unfortunate way of doing it, not only in terms of cost, but also there's a non-target effect, of that pesticide, that is sprayed, everywhere. The bees get affected, the other insects that are very useful in the ecosystem gets affected, and that's a problem. However, you can develop, a system that only releases, a certain amount of pesticide only when it's needed, and of course that can save you a lot of money and environment conservation. It's good for that. As I've said, no one took this project last year. Maybe one student can take a project on detection of, metabolites, for fruit ripening. That way, when the fruit is ripe, and so it can be picked and so forth. Maybe a student, can work on MIP, molecular imprinted sensors, for detection of this notorious pesticide, that is used especially in canola plants. And it's been found to be the reason why we have seen a decline in the bees population, because of its toxicity towards bees. And right now, we don't have a very good way of detecting, the use of neonicotinoids. And so if somebody can develop a sensor towards that, and, that can be interesting. And then lastly, detection of lactic acid. It's going to be interesting if somebody does that. Of course, you all know lactic acid is produced in sweat if at all you are under stress, from exercise. But also it's produced, by plants, and it can be actually an indicator, of fruit ripening as well. If at all a plant or a fruit still has a lot of lactic acid content, that means, of course, it's not very ready, so it can be used for things like grapes monitoring, if at all they are ripe and so forth. Yeah. And then somebody maybe can work on this, making a sensor that can be used to detect dissolved oxygen. And that way you can actually detect, if the plant is suffocating, possibly because of lack of oxygen, or if, you can use it also to detect dissolved oxygen in water, possibly in a fish, in aquaculture, where you're growing fish and you want to determine, the dissolved oxygen content, in that kind of an environment. So a very important, I would say, project. Now you're gonna see all these projects, sort of under one theme, and there's a reason why I decided to have one theme, because we can help each other. We can learn from each other. We can create that community of learners where somebody's work, can be used, by another person, and it can people can work together that way. If at all the themes, are very interrelated, but you can see the applications are equally very, very different as well. So hopefully, you guys are gonna make, that, you're gonna find something that's interesting for you, from this list of diverse projects. I should mention that in general, I actually recommend students coming up with their own research topics. However, this semester is very different, partly because of the pandemic. I anticipate, and I know this, ordering reagents, is very challenging. It's going to be slowed down. The supply chain has been substantially slowed down. And so if at all I've got so many different types of projects, that require ordering of reagents, I know I'm going to get into a problem. Not to mention, of course, you've got only so much limited time, only 3 lab sections, to actually make your project work. And so for that reason, I had to choose projects that I know for certain, that my research group, can really help you guys in to at least generate quick results, so that, you can write about it as well. And so for that reason, I propose this semester, students, stick to the list of topics, that I've selected, just because I know I've got everything that we need in the lab, to make it work for you. And again, we can help each other as well because the topics are very, very related. And again, that's going to help you guys because the likelihood of getting results that you can publish, where you can get a peer-reviewed journal articles, would be fairly substantial, in these projects. So as I mentioned to you guys before, please keep an eye on the deadlines, especially on the research project. Remember, you need to select the topic, and post it on Blackboard discussion board, by the 21st of January 2021. And of course, as an assignment, you get 1%, just by posting, your topic there. When you post your topic, again, I'm gonna assign you journal articles that you're gonna read, and then you're gonna present it, in class as your proposal presentation. Okay, and the proposal presentations, are gonna be, we already talked about it, last week, are gonna be on the 2nd of February, 2021. And of course, that's going to give you enough time to read and present it, present it in class. And of course, the idea, for you reading the journal articles is for you to design, sort of the protocols that you're going to need, to do your research topic. On the other hand, it also gives you, the background, or the literature review of the field so that you're well knowledgeable, on the field and the reason for the choices of, say, the reagents you use and the chemistry at play, and also the reason, for the analytical method that you're going to use and so forth. So again, choose a topic, by the 21st of January, I will assign you the articles, and then you can present, on the 2nd, of February. Now, this one you can take a review on it later, on how to write your research project, the term paper at the very end. But take note of this, I'm gonna require us first to write a draft that I can review for you and give you feedback so that you can improve on it, so that you can get better marks possibly, for your final thesis, at the very end. So that's all that I'm gonna say about the research project. Of course, if at all you've got questions, please let me know. As I said, I'm gonna make myself available every Tuesday and Thursdays during the designated lecture hours, for the office hours. Now, so let's start now, sort of the technical aspects, of analytical chemistry, instrumental analysis. Now, instrumental analysis is notorious, for using acronyms. And sometimes we just use acronyms and imagine it's common knowledge, to the audience. So I think it's important, for you, to be aware of the common acronyms, that are used, to describe instrumental methods. Because sometimes, as I said, you're just going to use them and assume you know what they are. For example, AES, stands for atomic emission spectrometer. DAD, which is a detector used a lot, with HPLC, stands for the diode array detector. It's a UV detector essentially. But some companies, like Agilent, among other vendors, they call it diode array detector. And you're going to talk about later on, of course, how it works and the components in it, and how it affects the figures of merit, such as the limit of detection and so forth. FTIR, I think most of us know about this, is the Fourier transform infrared, a very common instrumentation. MOL DMS, sort of this was my favorite, when I was doing my PhD, that's what I focused on, which stands for matrix assisted laser desorption ionization mass spectrometer. LDI, I think you guys would know that. It comes from this, laser desorption ionization. Electrospray ionization, the acronym is ESI. It's an ionization method, that is used in mass spectroscopy. FARB, fast atom bombardment, again, it's a ionization method used in mass spectroscopy. LCMS, I think most of us know about it, and you're going to do a lab on that in the lab, liquid chromatography mass spec. GCMS, that's a gas chromatography mass spec. Again, you guys, I think, yes, you used it last semester in Chem 211, and you're going to use it this semester as well. CEMS, means capillary electrophoresis mass spectrometer. Those of you in biology do a lot of electrophoresis, using the gel, but you can actually, make it into, put the gel sort of in the capillary, very similar to gas chromatography capillary, and then you do the, the separation that way instead of using a gel, on a platform. And so the CEMS, capillary electrophoresis mass spectroscopy, hopefully you're going to have time to focus on that technique. DESI, very new technique actually, possibly only developed around 2005 thereabout. Again, MOLDI was developed around 2000. So these ones are very, very new techniques actually. ESI developed again around 2000. The gentleman who did that actually got a Nobel Prize. And so forth. So they're very, very new techniques, but they've totally revolutionized, analytical chemistry, because they're really used, for detection, of proteins, which are expressed, by the genomes. And so they've totally revolutionized, the analysis. And they play a huge role to what people are calling, the development of drugs for personalized medicine, where people can profile somebody's genome, and then they can profile somebody's, proteins that are expressed, and try to link that, to clinical analysis. And so these techniques are used a lot for detection of proteins, more DMS, ESIMS, the DESI, and so forth. Chemical ionization, it's CI. Electron impact, it's EI. It's a component actually in GCMS. So the ionization method classically used in GCMS. Quadruple, that quadruple, triple quadruple, it's a component again in mass spectroscopy, which you're gonna come across as well. Other very common acronyms, TOF stands for time of flight, and you're gonna come across it in these in this course. QTOF, stands for quadruple time of flight, again as a component in mass spectrometer. APPI stands for atmospheric pressure photoionization. API stands for atmospheric pressure ionization. APCI, atmospheric pressure chemical ionization. PAD stands for photodiode array detector, and exactly the same as DAD, exactly the same thing. Okay, it's only that one company, I think it is the more scientific, they call theirs PAD, and Agilent calls theirs DAD, but they're exactly the same component. AAS stands for the atmospheric absorption spectrometer, or rather atomic absorption spectrometer. GFAA stands for graphite furnace AA, or atomic absorption. ICP is inductively coupled plasma. ICP-MS, inductively coupled plasma mass spectrometer. And this guy, the ICP-OES, which you guys are gonna use actually in the second lab, is it's inductively coupled plasma optical emission spectrometer. Now, of course, there are many others, that we're gonna come across, but I thought it's important, to highlight, some of these acronyms that I'm gonna be using sometimes in sentences, without necessarily defining them. And so it's expected, that you guys know about them. So just to summarize, our lecture today, again, we've already done this, post your research topics, and then after that, I gonna assign you journal articles, based on the research topics, and then you prepare a presentation that you will present to the class on February the 2nd on PowerPoint. Now, I also gonna post, additional notes with regard to how you should treat your lab notebook, which is part of GLP. GLP stands for Good Laboratory Practices. Laboratory practices. And all of us, are required, to follow GLP, which is a requirement in most of the industries that hopefully some of you are gonna end up in. And the lab notebook, is a major constituent of GLP, which mainly is on how well, you should document, your results in the lab. Now, considering, that you'll be doing a research topic, a research project as well, it now gets very, very close to what can happen to you in the industry in terms of, the requirement that you should detail, your results in a very accurate manner. Remember, some of the stuff that you detail in the lab notebook, could be, stuff that you can use in a patent, for example, if at all you're doing a research project that ends up, requiring to be, the technology requiring to be patented. And so the lab notebook in that case actually becomes a legal document that you can use to verify, that you're the first inventor. And so for that reason, very, very important as a classical core document in the good laboratory practices. And so I'm gonna post, a section of notes. This one was already covered in CHEM 211. So for you who did CHEM 211, there's someone gonna be a repeat. But for the other students, I think it's good you review what is required of you, for the good laboratory practices. And I'm gonna post that separately in a different video lecture, as well as I'll provide you the notes, the PowerPoint notes on that. Lastly, keep an eye on when the lab reports are due. Again, you've got this. So the first lab is gonna be due on that February and so on. So keep track on that, on the due dates, when each of those labs are gonna be due. All right, and talk again in the next lecture.